Hello guys, welcome to Ninja Explains.m. My name is Mubarak and I am your tutor.
Physiology Lecture 2. Blood. It is a connective tissue with a complex liquid intracellular matrix, plasma, in which cells or cell-like structures, formed elements, are suspended. The plasma is one of the ECF components, APRO3L in humans. The total blood volume in an average young adult male is approximately 8% of body weight. However, the volume of blood differ between species, breeds, individuals within a breed, and sexes, e.g., humans, male equals 5 to 6 L, female equals 4.5 to 5.5 L. Plasma, 55 to 57% of blood volume. Formed elements, 43 to 45%. Blood volume in various species. These may be about 15% lower in old or obese individual. FUNCTONS functions may be considered around two main activities. Transport. Regulation of homeostasis. Transport. Substances can be carried either dissolved and/or suspended to and from the tissues by the pumping action of the heart. The materials include amino acids, carbohydrates, lipids, minerals and vitamins from the gut as well as enzymes. Oxygen usually bound to RBCs from the lungs to the tissues and carbon dioxide from the tissues to the lungs. Heat from tissues to skin and lungs. Hormones from their site of production, endocrine glands, to target tissues. Excess body water and waste materials from tissues to appropriate organs for elimination. Homeostatic regulation. Regulation of volume and composition of interstitial fluids, ISF. Regulation of body fluid pH. Regulation of the body temperature. Protection against foreign invasion, antibodies, phagocytes. Protection against loss of blood, hemostasis. Components of blood. Blood settling either slowly or by centrifugation separates into two distinct layers. Firstly, the blood cells, mostly RBCs, form stacks like a pile of coins, rouleaux. The separation of blood results in the plasma overlying the formed elements and the white blood cells and the platelets forming buffy coat above the formed elements. This arrangement of blood cells is referred to as the packed cell volume, PCV, or hematocrit, HET. During slow settling or sedimentation, the time taken for distinct separation is known as electrolyte sedimentation rate, ESR. The ESR depends on several factors which include shape of the cells. Abnormal cells do not form rouleaux. Concentration of the plasma protein. High level of plasma protein tends to hasten rouleau formation. The presence of infections, toxins, anemia and other foreign material tends to increase ESR. In humans, normally, ESR 4. Newborns is about 2 mm per hour. Men 3, 7 mm per hour. Women 3, 15 mm per hour. White blood cells, leukocytes, sediment more slowly and form the buffy coat on top of the RBCs. The use of radioactive labeled plasma indicates that about 4% plasma remains trapped in the packed red cell volume. Therefore the HCT is an apparent one. Apparent HCT is approximately 43% to 45% of whole blood. Corrected HCT is therefore given by 0.96 times apparent HCT. The remaining straw-colored fluid is plasma, which contributes about 55 to 57%. Plasma, organic and inorganic. About 90% water, solvent, suspends and causes ionization of molecules and carries heat. Inorganic components, K+, Na+, Cl-, HCO3, etc., about 1%. Plasma proteins, about 7 to 9%. The plasma proteins, general functions, are primarily responsible for the osmotic return of filtered water. Contribute to the viscosity of blood. Create a suspension stability in the plasma that aids in maintaining dispersion of materials. Serve as reserve of amino acids, not used but available. Serve as buffers. Serve as antibodies. Aid in blood clotting. Plasma, organic and inorganic. There are three main fractions. Albumins, 55 to 64%. Globulins, 2%. Fibrinogen, 0.3%. Albumins 45G%, responsible for osmotic pressure of plasma, PCOP. Globulins, 2%. They are subdivided into alpha 510, 980 mg%, general functions. Beta 580, 1010 mg%, general functions. Gamma, immunoglobulins, 1200, 2150 mg%, and these are subdivided into types of gamma globulins, immunoglobulins. A. Constantly produced to help protect the respiratory and digestive tracts. It is only found in small amounts in the blood. Helps the immune system by preventing invading bacteria from being able to attach to surfaces in the body and from entering the bloodstream. It also helps to neutralize toxins and helps with the identification of invading bacteria. M. Produced the first time a particular bacteria, virus, or other antigen invades the body. It is instrumental in beginning the immune response to fight invading bacteria and viruses. G. This may be natural and acquired antibodies. It is the most numerous of the immunoglobulins. Produced by plasma cells as well. Functions to help the immune system identify invading bacteria, viruses, and fungi. D. Not known. E. Incomplete antibodies allergies. Fibrinogen 0.3%, 0.15 to 0.3%, responsible for blood clotting. Lipids found in blood. Fatty acids, 190, 420 mg%. Cholesterol, 150, 250 mg%, transit as fuel. Triacylglycerol, TAG, 20 mg%. Glucose, 60, 110 mg%. Enzymes, a wide variety acting on all categories of materials. Vitamins, A, D, E, K, B complex and C. Formed elements. 3 categories of formed elements in normal peripheral blood. Erythrocytes, RBCs. Leukocytes, WBCs, and thrombocytes, platelets. Erythrocytes, red blood cells. The mammalian RBCs are not nucleated. In camels and birds, the RBCs are nucleated. Mature red blood cells as they occur in bloodstream carry hemoglobin in circulation. They are biconcave non-nucleated discs averaging 6 microns in diameter in the bovine. The diameter of RBC is constant for a given species as follows. In dogs, the diameter is 7 microns. Bovines 6 microns. Goats 4 microns. Sheep 4.5 microns. Pigs, 6 microns. Man, 8 microns and birds 13 micron. RBCs are manufactured in the bone marrow. Numbers are higher in the adult male, ranges 4.5, 6m/mm3, than in the adult female, range 4.3, 5.5 m/mm3. Numbers are also dependent on age. The biconcavity of the RBCs gives it a large surface area of cell membrane per unit volume of cytoplasm. This ensures efficient diffusion across the cell membrane. The flexible nature of the cell membrane enables it to pass through the lumen of capillaries without rupture. Fate of RBCs. A mature RBC remains in the circulatory system for specific length of time based on the species of the animal. The lifespan of RBCs for different species of animals is as follows. Humans, 120 days. Horse, 140 to 150 days. Sheep, 45 to 55 days. Chicken, 35 to 45 days. As the RBCs become older, it does not show any structural changes. It becomes more fragile. the enzyme in the RBCs for metabolizing glucose and other substances, and for the utilization of oxygen gas becomes less active. Once the RBC membrane becomes fragile, it may rupture during its passage through tight spot in the circulatory system. Aged RBCs are removed from the circulatory system by phagocytic cells located mainly in the spleen, reticuloendothelial system, RES. When the spleen is removed, the number of abnormal and old cells in circulation increases considerably. The ruptured old RBCs are broken down into three parts, namely: iron portion can be used to produce more RBCs, or it can be stored in the liver. Protein part, globin, is further degraded into amino acid. Pigment part, heme, the yellowish, green pigment part is degraded into bile pigment in the liver, thus bilirubin and biliverdin. These are stored in the gall bladder as part of bile. When the liver is damaged or the bile duct is blocked, or when there is excessive destruction of RBCs, excess bilirubin spills over into the blood and stains visible mucous membranes such as the mouth and the eye. These therefore become yellow, a condition known as icterus, jaundice. Factors affecting RBC numbers: altitude, nutrition, prolonged exercise, sex of the animal, pathological conditions, age of the animal, erythropoiesis. During intrauterine life, the following serve as sites for RBC production: yolk sac, spleen, liver, lymph nodes and red bone marrow. At or shortly before birth, all areas cease production of RBCs except the red bone marrow in such areas as the sternum, skull bone, vertebrae, ribs and the ends of long bones. The cells originate from a stem cell common to all formed elements, and pass through a series of stages to form the mature cell. Hemoglobin, a red pigment, accumulates during the later stage of development. Many substances are required for the synthesis of RBCs. The formation of RBC is inhibited by a rise in the circulation levels of RBCs to supernormal values, and to be stimulated by anemia. It is also stimulated by hypoxia. One important factor controlling RBC synthesis is the glycoprotein known as erythropoietin. The kidney, in response to hypoxemia, produces renal erythropoietic factor, REF, which act on a plasma globulin precursor to form erythropoietin. Erythropoietin stimulates mitosis of marrow stem cells and their maturation to RBCs. The following are also required for RBC production: lipids, cholesterol and phospholipids, incorporated to membranes and stroma. Protein, incorporated into cell membrane. Iron, incorporated into hemoglobin. Amino acids, incorporated into hemoglobin. Vitamin B12 used in DNA synthesis. Intrinsic factor, factor responsible for the absorption of B12. Pyridoxine, B6 increase the rate of cell division. Folic acid, promotes DNA synthesis. Cobalt and copper, serve as catalyst in hemoglobin synthesis. Functions of RBCs. Transport of oxygen and some carbon dioxide depends on the presence of the respiratory pigment hemoglobin in the erythrocytes. HB increases the ability of the blood to carry oxygen by 60-fold. 13 to 16 g of HB per 100 milliliters of blood represents a normal amount. HB is also a good buffering compound because it is a conjugated protein. The erythrocytes contain potassium bicarbonate for buffering purposes. The presence of carbonic anhydrase in the RBCs speeds up the conversion of carbonic acid to carbon dioxide and water. Assignment: write essays on HB, abnormal HB, destruction of HB in jaundice. Abnormalities in the erythrocytes formation. In the formation of RBC, various processes may result in abnormalities in the shape of cells, poikilocytosis. Hemorrhage, heavy exercise, and exposure may result in a release into the bloodstream of immature erythrocytes that are larger than the normal and that contain nuclei or fragment of nuclei. abnormal size, anisocytosis or numbers of cells, polycythemia or anemia. Polycythemia is an increase in number of RBCs to greater than normal for the species. This may be a response to decreased oxygen content in the air that is breathed, caused by the release of erythropoietin, and is called physiological polycythemia. Pathologically, this can occur from tumor-like conditions in the bone marrow. In such conditions, the blood flows very sluggishly because of the increased viscosity. Oxygen extraction from the cells is much increased, and the tendency to show a bluish color to the skin, cyanosis, is increased. Red cell fragility. RBCs, like other cells, shrink in hypertonic solutions. In hypotonic solutions, they swell and become spherical, and eventually they lose their HB. Hemolysis dissolves in blood, coloring it red. With normal osmotic fragility, the RBCs begins to hemolyze when suspended in a 0.5% saline. 50% lysis occurs in 0.40 to 0.42% saline, and lysis is complete in less than 0.4% saline. Anemia. A deficiency of RBCs in the blood. Physiologically, there are four major types. Caused by blood loss. Caused by bone marrow aplasia or hypoplasia. Caused by maturation failure, and caused by hemolysis.